Okay, now, that cell division has to be tightly controlled. There are some checkpoints. These checkpoints are going to occur in some very specific areas of the cell cycle. One of the checkpoints is going to be in the G1 phase. In the G1 phase, you're going to have a checkpoint there that checks for the new cell, that new cell has a DNA that is clean, that has no problem. So in that checkpoint, the cell is going to check for, let me make sure that I'm telling you the right thing, that the cell is going to check for the integrity of DNA. If DNA is damaged in this new cell, that cell has to die. Okay? But if DNA is not damaged, then that the cell has to be allowed to go on and do the job it is designed to do in the body, right? So after DNA synthesis, you are going to have another checkpoint. That checkpoint is also going to be a DNA damage checkpoint, right? Because during synthesis, not all synthesis is going to end up with a clean DNA. So anything that is deemed not normal is going to cause the cell to undergo apoptosis. If the DNA is damaged, that cell has to stop cell division, okay? And if the repair can be made, that repair will have to be done. If that repair cannot be made, that cell will have to die by apoptosis. And then you have the spindle assembly checkpoint. The spindle assembly checkpoint here is going to be a checkpoint where the cell is going to make sure that each centromer has a mitotic spindle attached to it, because that attachment is what is going to guarantee an equal pool of the chromosomes so that they're going to line up on a quarter line. That's one. Two, that as the mitotic spindle are pulling, that the two sister chromatids are going to come apart, and that each copy is going to move toward opposite direction, right? If some of the chromosomes do not have mitotic spindles attached to the centromers, then this is going to stop. Okay? If that doesn't stop, then the cell will have to undergo apoptosis, right? So there is a tight control during the cell cycle. One, around integrity of DNA, that the DNA was properly copied, right? Two, that the mitotic spindles are attached properly, right? Three, that the new cells have a genetic information that is clean in terms of quantity, but also in terms of quality, right? Because let's face it, the mitotic spindle may not be attached to some chromosomes here, and the cell cycle can can also continue, causing one daughter cell to have too many cells, too many chromosomes, and another daughter cells having too little, right? So when the cell is controlling this during the G1 control, it's going to notice that, oh, this cell has too many chromosomes. That cell needs to die. This other cell over there, it has too little chromosomes. That cell needs to die, okay? So during the cell cycle, you have a number of elements that are going to play a role in controlling how many times the cells are dividing and when the cell needs to, stop dividing. These elements are, for instance, the telomeres and the growth factors. But let's start with the telomerases, the telomers. The telomers occur at the end of chromosomes. So let's say this is one chromosome, and that one chromosome is going to have some fragments of DNA that are repeats of sequences of nucleotides. And the sequences of nucleotide are 6 nucleotide, T, T, A, G, G, G. This T, T, A, G, G, G can be repeated several times on a chromosome, right? At the end of a chromosome, what happens is that each cycle, whenever a cell divides, you have some fragment that is lost here. That's first division. Second division, some is going to be lost. Another division, some is going to be lost. At some point, there is going to be a number of division after which these telomeres do not exist anymore. And now the cell is starting to attack the very information that is the core information that will cause a cell to be healthy, right? So when that happens, that cell is going to die. So now, each division, a cell is losing roughly 50 to 200 nucleotides at the very end of its chromosomes. So the telomeres are shortening, right? That shortening is going to limit the number of times a cell can multiply, right? Which, when we consider a standard cell is going to undergo about 50 divisions before the telomeres are in such way that the very core information is now being attacked, and that cell has to be tagged for death, right? And in general, it's going to be programmed cell death. Now, some cells actually see their telomerases not shrinking. Okay, this is the case for sperm cells, egg cells, cells in the bone marrow, which are, by the way, stem cells, and cancer cells. Now, cancer cells, they are able to actually produce that telomerase, right? And the job of the telomerase, it's instead of seeing that telomere shrinking, right? It's actually going to grow the telomere so that this telomere never really shrink to the point that the cell will stop dividing after so many divisions, right? Now, other components that can affect cell division are the hormones. The hormones are going to control cell division in the sense that they can speed up cell division. They can also decrease the frequency of cell division. We have some growth hormones, right? These growth hormones are responsible for speeding up cell division. Then we have growth factors. The growth factors, for instance, one particular growth factor I want to mention is the epidermal growth factor, the EGF. The EGF are essential for causing cells to multiply. Now, think about your skin cells. Your skin cells multiply all the time because your skin sheds all the time, right? Your skin cells are growing all the time, and this is possible because of the epidermal growth factors, right? This allows to stimulate cell division in the skin, right? By the same token, when the skin does not need to multiply, say for instance you have a cut, that cut gets repaired. And after the cut is closed and completely repaired, then what is the purpose for further cell division, right? the cell has to stop cell division, and you have some factors that are going to actually stand on the way of cell division, and this is where the checkpoints come into play, and the control of the cell cycle becomes important. Now you have two major components that are responsible for the tight control of the cell cycle. You have proteins that are called cyclins, and you have other proteins that are called cyclin-dependent kinases. These are enzymes. These enzymes, actually what they do is that they transfer a phosphate to other chemicals, and by transferring a phosphate to other chemical, they either stimulate cell division or they shut down cell division. Now, the cyclins are essential here because without the cyclins, the cyclin-dependent kinases are not going to do their job. So these kinases, CDK, have to be associated with cyclins to be able to actually do their job. And their job is going to be about, one, either stimulating cell division or shutting off cell division. And this is how the body is able to control when the cell needs to divide and when it needs to stop divide. Now let's switch our gears to apoptosis. Apoptosis is going to be a programmed cell death, what I call clean death. You know, for instance, I was telling you about the web between fingers, right? And the ability to actually have perfectly designed fingers because of the cells between fingers that are going to die in a designed manner, right? That clean death is essential for the integrity of the body. But also, you do have some type of deaths that are not because of a clean death, but that are because of a damage. When that happens, the cells undergo what we call necrosis. So necrosis, it's a death that is because of damages. For instance, when you consider when the skin is actually exposed to a huge amount of UV, the skin cells that are damaged are going to die by necrosis, as opposition to a clean death that is apoptosis. So here we are going to have some enzymes called caspases. These caspases are going to be essential to trigger programmed cell death. The caspases are going to trigger programmed cell death so that the cell is going to get their cytoskeleton completely disorganized. And as the cytoskeleton is disorganized, the lysosomes will start now eating the cells from the inside. And as they eat the cell from the inside, these cells are going to shrink in size, and eventually these cells are going to be engulfed by phagocytic cells, including macrophages. Let me write it down here. Macrophages, okay, to discard the damaged cells. Now, let's talk about stem cells. Stem cells occur in many kinds based on their origin. Now, a stem cell is a cell that is a blank cell. That blank cell can multiply over and over and over. We call this self-renewal, right? Stem cells have to be able to renew over and over and over so that you always have a batch of blank cells. Now, some cells, though, rather than renewing, are going to undergo a process that we call differentiation. And that differentiation depends upon the signal they receive. Some signals are going to cause the cell to undergo the pathway where they become skin cells. Others are going to undergo the pathway where they become blood cells. Even among the blood cells, you have red blood cells and you have white blood cells. And in the white blood cell, you have different kind of white blood cells, right? So these cells that undergo differentiation, before they are completely differentiated, they are going to become what we call progenitor cells. These progenitor cells are blank cells, but that are already predetermined to become a particular type of cell. For instance, derivatives of stem cells in the bone marrow are designed to become blood cells. And these blood cells can become red blood cells or white blood cells. Now, in the white blood cells, you have, for instance, the monocytes, you have the neutrophils, to mention only these two. You have the basophils. I'm mentioning a third one. In any case, these progenitor cells are cells that are designed to become white blood cells in this particular instance here. Okay? Now, it is very important that you always have progenitors of white blood cells and progenitors of red blood cells and progenitors of skin cells and so on and so forth, so that the body doesn't have to rely on the blank cells that still have to go through the entire process of differentiation. This progenitor cell is only a step away from becoming the final differentiated cell, right? Very important to know. Now, the stem cells, I was mentioning that they occur in many kinds. Some stem cells are what we call totipotent cells. These totipotent cells can give rise to about any cell. They can give rise to skin cells, muscle cells, nerve cell, you name it. These are the kind of cells that you are going to find during the development of an embryo, right? And these stem cells that can become about any cell is going to partially differentiate, some of them at least, are going to partially differentiate to become pluripotent cells. These pluripotent cells are cells that are still stem cells, but they are already partially differentiated to only differentiate into a limited number of type of cells. For instance, you have your stem cells in your bone marrow, right? These stem cells are pluripotent cells because they can become red blood cells, they can become white blood cells. And in the white blood cell, you have different kind of white blood cells, right? So then we have the multipotent cells. The multipotent cell, they only have a few developmental choices, okay? They can only become one or the other, okay? So this stem cells, they are not all present in adult cells. At least the totipotent cells are not anymore very much present in adult tissues, right? While they are still very present in an embryo, right? The multipotent cells are very much present in adult tissue, okay? So here, I'm just showing you how you go from a sperm and an egg to form a fertilized cell that we call a zygote. That zygote is going to multiply to give you some stem cells. These cells are totipotent cells, totipotent stem cells, okay? These cells have to be able to replenish, right? And to differentiate somewhat into progenitor cells. These progenitor cells themselves have to multiply and differentiate into some other progenitor cells. Now, the progenitor cell right here is not the same as the one right here. Everything depends on the signal they received to become the progenitor cell they are. And now, if we focus on this particular progenitor cell right here, that progenitor cell is going to develop into somewhat differentiated cell here, a future fibroblast, and that is going to differentiate into a fibroblast, that is the specialized cell in a connective tissue. And eventually, you have this progenitor cell that differentiate into this other progenitor cell, and that progenitor cell can multiply and eventually differentiate into bone cell. And notice that a bone cell is not a fibroblast. They don't exist in the same kind of matrix, right? Some cells, now on this side right here, some cells are going to become progenitor cells to become red blood cells and white blood cells and platelets and so on and so forth. Now, on the other hand, if we actually move this direction, right, the progenitor cells here are going to evolve into other progenitor cells, some going toward forming skin cells, others going toward forming nerve cells, others going toward forming supportive structure of nerve cells, including astrocyte. So this is just to explain the complexity, right, of stem cells and as they go from totipotent cells to pluripotent cells to multipotent cells. Now here the slide actually shows you the origin of the different stem cells. You have some cells that are embryonic stem cells. These are the cells that are totipotent cells, right? And they are very much, they can differentiate into about anything. So these cells are very important for research in the production of new organs, right? We have the induced pluripotent cells, which is actually done in a lab where we're going to use chemicals to actually cause some cells to go from specialized to non-specialized, so that you can actually produce whatever kind of cell you want. And then you have the adult stem cells. These adult stem cells are, for instance, the cells in the red bone marrow. Okay. All right, so what do we use stem cells for? We can use stem cells for many purposes, right? We can have a donor cell here that is going to go toward solving a health issue, right? We can have this individual here that can provide their own cells to actually address a problem in their own body. You can have, or let me come back here. Look, you can have a person who is healthy, who is going to donate their cells to actually solve a problem in an individual who is sick. You can have some somatic cells in an adult individual, in an adult individuals. These somatic cells, stem cells, are submitted to chemicals so that these cells are reprogrammed to actually become stem cells. And these stem cells can now be cultured in a plate. And as they are cultured in a plate, they are going to be given a particular signal based on the kind of tissue we want to produce. We can cause this stem cell to differentiate into blood cells, into muscle cells, and into nerve cells. Everything depends on what we want to do. So the stem cells are used for many purposes. I already mentioned that, but it is worth asking the question, okay, well, how about ethics? How should we use these stem cells, right? What is the origin of the stem cells? If we obtain stem cells from adults, okay, well, we can assume that, well, that adult is giving a consent, right? How about embryonic stem cells? When we talk about embryonic stem cell, who is giving a consent? Parents are giving consent, that's true, okay? But the embryo is an embryo, an organism, right? If we define an embryo as an organism, can we then say that, well, if we are using embryonic stem cells, who are we asking? Are we asking that very embryonic being, their consent? Or, I'm just, try to draw out there that there are some questions. And finally, in terms of ethics, what are we doing with the information that we obtain from culturing stem cells? Where are we storing that information, right? Can this constitute a threat, right? These are questions that we don't always think about, that are worth asking. There is no right or wrong question. There is just a matter of, trying to make sure that in terms of ethics, we are keeping good practices. Finally, we're going to close this chapter with the human biome. What do we call the human biome? The human biome, it's all the microorganisms that are found in you. Found in you, found on you, within you. All these microorganisms constitute your microbiome. And that microbiome is going to vary tremendously throughout your life. What is happening in the childhood is not necessarily happening when you are a toddler, when you are an adult, when you are an elderly. Now, an unborn individual is completely free of microorganisms, right? But that organism, when it comes to life, through birth channel or through C-section, this is when they start getting exposed to some microorganism, and now they can now start building their own microbiome. And as studies show, a child who is actually now crawling is going to have a different microbiome based on their diet. For instance, a child that is being breastfed is not going to have the same microbiome composition as a child that is fed with formula. Okay? And when a child starts eating solid food, their microbiome also changes over time. When an individual is a toddler, they start walking. They start doing something they didn't do when they were babies, right? Okay, so these individuals are now going to have a different microbiome based on their condition. If they were treated for an infection, they will have a profile of microbiome that is going to be different compared to if this individual were malnourished, okay? Compared to if these individuals are healthy. When an individual becomes adult, their microbiome changes as well, right? We look at the composition of the microbiome in a healthy individual as opposition to an individual who is obese, right? The composition is different, okay? When an individual becomes old, we also see a different composition of the microbiome, whether this individual is actually 65 up to 80 or more than 100 years old. we see that variation. So that variation is based on many aspects. So our body is filled with microorganisms. We know today that some microorganisms are responsible, for instance, of the development of acne in individual, right? We know that circumcision is actually modifying the microbial flora so that individuals who are not circumcised are much more prone to infections such as HIV infection, okay? And there are other that have been associated with some diseases, including Crohn's diseases, including some cancers, basically based on our lifestyle. The microbiome is different from one individual to another. Now, we know more and more that actually our health, whether we have dementia, is now more and more associated with our microbiome, right? Whether you have mental issues, whether you have dietary issues, and many other chronic diseases such as diabetes, obesity, cardiovascular diseases are now more and more associated with actually some health conditions. So this is where we are going to stop our conversation. Thank you for your attention. Bye now.